Hi, good morning, and welcome to the FoodSense webinar. So we do this Q and A webinar every week at approximately 8 a.m. London time. We record it, we put it online, so we try to answer questions that have come in during the week, specifically around our FoodSense technology. Now, one of the questions that's come in this week is really about repeatability. Now, I've put accuracy, precision, and repeatability here because people really sort of sometimes mix up these terms that really, I think often people just want repeatability, that, that they take the same sample and they measure it and they get the same result again. So I will talk about that today. And they also want to know with FoodSense, because capsaicin, which is the molecule, so for example, if you're measuring chili, the molecule that gives you the perception of hotness or the sensation of hotness is a molecule called capsaicin. Capsaicin is not too super soluble, and so actually we have to change the volume of dilution based on how hot we think the product actually is, and I will discuss that today as well. So as I say, this is the 16th of August. It's approximately 8 a.m. London time, and this is our weekly Q and A session on FoodSense. Just to say as well that we do have a webinar on the 29th of August, answering questions, and I'm doing a live demo on FoodSense on the 29th of August, so hopefully you can come to that as well. Talking about accuracy and repeatability. So I've got a video playing here of one of our scientists called Solrun, and in this one, this is a good example of where this, I wouldn't say issue or concern with accuracy and repeatability comes from. Because in this video, Solrun takes a chili, which I would describe as a very heterogeneous product. That it means it's got seeds, it's got pulp, it's got skin. The capsaicin is not evenly distributed in these chilies. So in this video, she's actually prepared chilies by blending them all together. So it's a very heterogeneous sample. So that's the first, can I call it, issue that comes about, that actually chili samples themselves are not super homogeneous, they're heterogeneous. Okay, now the other thing that happens is people get these certificates of analysis from HPLC labs. You can do this experiment for yourself, but if you send the same sample to 3 different HPLC labs, you'll get 3 different results. I can absolutely guarantee you that. None of which are completely the same. So there's a repeatability problem, let's say, with HPLC testing. There's another test that the larger developers of chili products have done, where actually they've sent the same sample to the same lab, but on different occasions. And again, they would actually get different results. So be careful with the idea that HPLC is some sort of gold standard, because it is not. The repeatability from these labs is not the same. Now, what I want to do here is describe the idea that I've got a chili product and I tested it on my FoodSense, and the first result I got back is 17,000 Scoville heat units. I took another sample I tested, I got 22,000 Scoville heat units. I took another sample I tested, I got 20,000 Scoville heat units. I had another sample I tested, I got 15,000 Scoville heat units. I took another sample I tested, I got 21,000 Scoville heat units. So the range on this is, the minimum is 17,000 and the maximum is 22,000. So there's quite a large range there. That's 5,000 Scoville heat unit range. The average of this is 19,000 Scoville heat units. The standard deviation is 29,015. It's not 29,000, sorry, 2,915. And then the coefficient of variation for this series of tests is 15%. For a natural product that's very heterogeneous, and I'm testing 5 samples, this is actually a good score. That if I can call it, the coefficient of variation tells you something about the sort of spread in the data, but it's also a sort of indicator of repeatability. So this 15% is actually not bad. And the 15% just comes from the standard deviation divided by the average, and I've turned it in percentage. So what I want to say is, if you're concerned about your repeatability, then send us your results, but we will need multiple results so that we can do a statistical analysis on your results. So this is sometimes a fairly standard thing. We find that people do have these different results, but when we then do an average and a standard deviation and we look at this coefficient of variation, we actually find out that they're actually pretty good. So we can do some basic statistics on it and find out, how repeatable this really is. And then we do have people who are using this technology in process control, and this is what they're doing. They're doing something like 3 tests, doing an average, doing a standard deviation, making sure the standard deviation is not too big, making sure it's small enough. And if it's small enough, then they think the tests are repeatable, and then they can rely on the average result. Now at this point, we're on our generation 3 of the food sensors. It's been around quite a while. And things that we've sort of discovered over the years is that capsaicin is not terribly soluble. And so what we find out there is if it's mild, a mild material, a mild source, for example, we can use a very small volume. But as the material becomes hotter with capsaicin, we have to use a larger volume of material. And this is because capsaicin, it's between 13 parts per million and 29 parts per million soluble in aqueous solution. So the reason that you get these hotter sources and etc. is because of the oils, and some of them are called lipids. But it's the extra materials in the chili that help to solubilize the capsaicin. But intrinsically, capsaicin is not terribly soluble. So what we're doing is, if we think it's mild, something like 10,000 Scoville heat units, we tell people to do a 1 in 10 dilution. If it's more medium, something like 100,000 Scoville heat units, we'll tell them to do a 1 in 100 dilution. And if it's really hot, and I want to correct this now actually, if it's really hot, we will tell people to do, and I sort of say greater than 250 Scoville heat units, 250,000 Scoville heat units. Let's just correct that very quickly. Then we would tell them to do something like a 1 in 1,000 Scoville heat units, which is, sounds a little, I don't want to say extreme, but it's just because capsaicin itself is so terribly insoluble. So what I want to do is then is just say that we do have a webinar. It's on the 29th of August. We do have another webinar booked on the 26th of September. If you can make it, that'd be great. And you can see that what we're doing in these webinars is, these weekly webinars, I should say, is answer questions. So this week, the question was repeatability. So if you want us to look at repeatability, send us something like 5 tests on the same sample, and we will do the statistical analysis for you. How do you know what volume of dilution to do? If it's mild, something like 10,000 Scoville heat units, which I think a lot of sources are in that kind of range, doing 1 in 10. If it's more intense, sometimes like chili powder, do a 1 in 100. That's about 100,000 Scoville heat units. If it's really hot, do a 1 in 1,000, but that really rarely happens actually. I'd rather you were playing with or using 1 in 10 and 1 in 100. If you've got any questions for us, don't hesitate to reach out to us. And the next Q and A on this, the date will be wrong on this, but the next Q and A for this will be next week, which will be the 23rd of August. So if you've got any questions, don't hesitate to reach out to us. Okay, thanks very much.